What's up, AP Bio students? This is Mr. McLeod again. Welcome to our series of podcasts on cellular transport. In this series, we'll talk about how substances get in and out of cells and the methods cells use to move important materials. So get your notes out, something to write with, and let's transport. First, let's review some cell membrane basics that you learned in your biology class. Most cellular membranes are composed of phospholipids. You see the word right here, phospholipids, meaning there is a phosphate component and a lipid or fat component. That's why they call them phospholipids. First, let's get a basic drawing of a phospholipid in your notes. That's this structure right here. So let's get this in your notes, this picture of a phospholipid, and then label it as a phosphate and then a fatty acid tail. So phosphate heads, fatty acid tails. So go ahead and get that diagram in your notes right now. So now in your notes, write that the phosphate head is hydrophilic, and that hydro means water, and philic means loving. So also write that the phosphate head attracts water molecules. You see that written right here. So the phosphate heads are hydrophilic. There's the word, hydrophilic, which means they attract water. Then write that the fatty acid tail is hydrophobic, and that hydro means water and phobic means fearing. And also write that the fatty acid tails repel water. Next, let's have you get the structure diagram of a typical membrane in your notes. That's this one right here. You got to recognize this diagram as a phospholipid membrane. Now we're going to call it a bilayer as well. Bi meaning two, so there's two layers of these phospholipid heads and tails, kind of tail to tail from each other. So the membrane has depth. It doesn't look like it because it's really small on a microscope, but it's got thickness. Now you see here how these are arranged with the heads on the outside and the tails pointing toward the middle. This is important because the heads attract water into the cell and the tails help repel water out of it. So make sure you get that down as well. Now you also need to understand why this is referred to as a bilayer. So can you remember why? We just said it a couple seconds ago. Remember the prefix bi means two, so if you look at the membrane structure, it has thickness because of the two separate top and bottom layers that you see here that should now be in your notes in a diagram. Okay, now let's look at this diagram of a bilayer in a little more detail and get some of the details into your notes. First of all, add to your notes that the hydrophilic heads are also polar. Do you remember what that means? If you said that it's polar because it has a positive and negative end, you'd be correct. We've always used water as an example. Also write that because these heads are polar, they attract other charged ions or polar molecules. Now this allows substances like sugars, water, salt, and waste to pass in and out of the membrane relatively freely. Then write in your notes that the hydrophobic tails are nonpolar, meaning that they do not have charged ends to attract other substances with. And that because of this structure, polar molecules cannot pass through these hydrophobic tails at all. This will become important when we start to describe the movement of molecules in and out of the cell through this membrane. Now, last thing, get in your notes that because the membrane does not allow all substances to pass through it freely, it's said to be selectively permeable, which means it kind of selects which molecules gets in and out and which ones don't. Okay, now let's just mention some important functions of the cell membrane for review purposes and get them in your notes. First, the cell membrane separates the cell from the environment it lives in. We call this environment its aqueous environment. A typical cell is somewhere about 8 nanometers thick. We use the thick word kind of loosely there. It's actually really thin. But remember, it's got that membrane's got thickness. You see it here again. Next function is what we just mentioned. The membrane controls what gets in and out of the cell. Some substances like water cross easily, while other like fats do not. Okay, next, write in your notes that polar substances like water pass easily through the membrane while nonpolar substances need help, and that one of the ways the membrane provides help is through the use of what we call protein channels. And you see some drawings of protein channels. Here's kind of an artist's rendition, and here's more of what proteins look like. You recognize the structure of them, the primary, secondary, tertiary, and quaternary structures. So these are kind of a basic look. These are more of a realistic look of what these proteins look like. Okay, so also write that protein channels allow specific materials to cross the membrane that otherwise couldn't. You can see here that protein channels are actually embedded, that we're going to see that word a lot, embedded into the cell membrane. Now, they're not a part of the cell membrane, they're just embedded in it. So it kind of makes them part of it, but they're not part of this structure here is what I mean to say. Also notice that these channels connect the inside and the outside of the cell and are very specific to the substances they help get through the membrane. That should be no surprise. Structure and function, right? Different structure, different function. Next, get in your notes that the cell membrane has more than just lipids and phosphates. It also has what are called transmembrane proteins. You see that right here. Transmembrane proteins that are embedded in the phospholipid bilayer. And these transmembrane proteins are semi-permeable channels that also allow substances to move in and out of the membrane. We'll look at how these work in an upcoming podcast. So again, these create these semi-permeable channels and are just slightly different now than the proteins we mentioned in the previous slide. The membrane's very complicated, much more than you probably imagined. Okay, can you answer the penguin's question? So he's asking, why are proteins the perfect molecule to build structures in the membranes? If you said that because proteins are made up of different amino acids and that each amino acid has different properties, such as being hydrophobic, hydrophilic, acidic, basic, etc., you'd be correct. Also write that proteins can basically bind to almost anything, but different ones will bind better or worse with particular structures based on the composition of amino acids. So it's the complex structure that really makes proteins the perfect molecule. So they can actually be made to do almost anything. And remember our discussion of enzymes, which are the most specialized proteins. So you remember that proteins are probably the most important of the organic molecules. This is no exception. Okay, now let's talk about something called signal transduction. Signal transduction. Write that in your notes. Then write that signal transduction is transmitting a signal from outside the cell to the cell nucleus. And then an example would be like receiving a hormone which triggers a receptor on the outside of the cell that then signals the cell or signals to the nucleus that a protein must be made. Remember the whole protein synthesis thing? So there are really 6 functions I want you to get in your notes for membrane proteins. You see them here in this slide. Just list them, and then eventually we're going to go over them. So get them all in your notes now. We'll talk in more detail about each as we go along. So there's transporter, enzyme activity, cell surface receptor, cell surface identity marker. That's what that says. I think it might be cut off on your screen. Cell adhesion and attachment to the cytoskeleton. That one might have got cut out too. Sorry about that. So this one is cell surface identity marker. We're going to talk quickly about that one here in the next few slides. And this one is attachment to the cytoskeleton. So get those 6 listed in your notes. Now, it should come as no surprise that a protein's structure determines its function in the cell membrane. Now, write that the cell membrane and the membranes of all the organelles, or it's probably escapes you, but organelles also have membranes. That each of them have a unique collection of proteins, meaning they each have a different function. So there are 2 main classes of membrane proteins you need to get in your notes. First are the peripheral proteins, which are loosely bound to the surface of the membrane. Now also write that an example of a peripheral protein would be an antigen. You saw it introduced in the previous slide. Also write that antigens are referred to as cell surface identity markers. So there's a great example of one right here, a little pictorial right here, of an antigen or a cell surface marker. Now this is very important to your immune system. It needs to know which cells are yours and which are foreign, so it knows which cells to attack. Now the second class of membrane proteins we want you to get in your notes are the integral proteins, which penetrate the lipid bilayer and stretch across the whole membrane. And you see those right down here in the corner. Examples are transport proteins, which you diagrammed earlier. So peripheral proteins, integral proteins. So those are the 2 classes. And you got to know an example of each. Now something historical, just because I know I've seen it on an AP test in the past, may not have been in the past couple of years, but let's just get it in your notes. Write in your notes that in 1972, S.J. Singer and G. Nicholson proposed that membrane proteins are actually inserted into the phospholipid bilayer after that layer is formed, and that this model is referred to as the fluid mosaic model. Right there. Fluid mosaic model. So get that in your notes, 1972, Singer and Nicholson. So here's a great example of what they proposed. A lot going on here. They didn't know everything here, but this is now a picture of the fluid mosaic model that they proposed. So again, much more complicated, a lot more going on in the membrane than people ever thought of before. So just a diagram for you to recognize in case you see it. Okay. Next, let's get in your notes that the cell membrane also contains carbohydrates, but that these carbohydrates are not inserted into the membrane. They are too hydrophilic for that. They are actually attached to embedded proteins, and those are referred to as glycoproteins. And you see one right here. Again, we're going to talk about cell recognition. This is an antigen now attached to an embedded protein. So we call glyco, remember a carbohydrate, or glycogen is a carbohydrate, a glycoprotein. Now, also write that these glycoproteins called antigens play a key role in cell-to-cell recognition. We've mentioned that already. Now, the 4 human blood groups are an example, A, B, AB, and O. So I've seen that on an exam as well. So they want you to give an example of an antigen, and the blood groups A, B, AB, and O, which we'll talk about later in genetics, are an example. Now, those differ in the external carbohydrates, for example, on red blood cells. Get in your notes that these glycoproteins are important in organ and tissue development, and also are the basis for rejection of foreign cells by the immune system. Okay. That's it for part 1. So take a picture of those notes, submit them to Moodle and grab that study guide, answer the questions, and I'll see you in part 2. Bye.